Hi, I'm Bob Naviaux. I'm going to be talking to you today about our work with metabolomics and exposomics of acute Lyme disease. This is just a brief 8-minute summary of our work. To introduce you to metabolomics and exposomics, we think of drawing a sample of blood as taking a sample of water from a river or ocean ecosystem that contains all of the nutrients and the waste products that are produced by all the cells of the body, including all the microbial cells in the body. And so that sample can be considered a little microcosm, and the endogenous molecules consist of comprise the metabolomics, and that is analyzed through the mass spectrometer. And then the exogenous molecules, those collection of molecules are called the exposome, and we analyze those through another mass spectrometer in order to get an idea of toxicants that may be involved with human disease. So this is an overview of the metabolomics and exposomics platforms that we use at UCSD. So we analyze about 690 naturally occurring molecules from a drop of blood that include over 60 different biochemical pathways, for example, phospholipids and purines and sphingolipids. And then in the exposome, these are all exogenous molecules. We analyze 1,232 molecules from using both gas chromatography, tandem mass spectrometry, and liquid chromatography, tandem mass spectrometry. And we look at pesticides and flame retardants and plasticizers, phthalates, persistent organic pollutants like PCBs and bisphenol A, as well as mycotoxins and a variety of cyanobacterial biotoxins, food additives, and flame retardants. So in this study that I'll present today, I'll talk about our results from 207 of the 1,232 chemicals. So to give you an overall outline of the work, the subset of the work is from samples that were collected by the Bay Area Lyme Foundation Biobank. We analyzed, eventually we'll analyze about 2,000 chemicals, but we have a group of 60 females and 66 males. They're divided into females with acute Lyme, and then paired convalescent samples, and then regional controls, and then the same for males. And I'll be talking about today just the results for the females. And the two different areas that we're talking about are the Lyme hotspots centered roughly on Long Island, New York, and then the second one in the upper Midwest in Wisconsin. So you can see they're about 2/3 from New York and about 1/3 from Wisconsin. The first thing that comes out when you look at the data of all the samples is that, interestingly, the metabolomes of all the males segregate together and all the females segregate together. So there is a overlapping but non-identical metabolic response to acute Lyme in males and females. I'll just talk about the results from females today. So metabolomics separates the acute Lyme samples from regional controls. And then when you look at the pathways that are most changed in the acute samples, you see a mixed picture of certain phospholipids increase and others decrease. But they're glycosphingolipids that include gangliosides with sialic acid residues that are used actually for receptor binding by a number of different viruses and microbes are downregulated. And then some of the non-sialoglycosphingolipids are increased. There's an increase in carnosine and histidine. Interestingly, we saw a 15% increase in thyroxine and a decrease in pyrophosphate, which is a natural inhibitor of calcium deposition in arteries and arterioles. there was a decrease in a key metabolite of cholesterol synthesis called IPP, or isopentenyl pyrophosphate. Also a decrease in the precursor to vitamin D that is in the skin called 7-dehydrocholesterol. While there was an increase in aldosterone represented, which we see as a reflection of autonomic dysregulation that occurs with acute Lyme. Other molecules like creatine were decreased, but methylcysteine was increased. And adenosine, an important final metabolite of ATP catabolism, was decreased. And down at the bottom here, you see that the activated 1,25-dihydroxyvitamin D3 was decreased. Now, an important observation from analyzing the convalescent samples was that they do not return to the pre-baseline health condition. And we still see them as significantly different, and that there's a very dramatic decrease in a class of sphingolipids called ceramides, which is something that we also saw in our previous studies of chronic fatigue syndrome. The endocannabinoid, anandamide, and another endocannabinoid called N-oleoyl ethanolamide, or NOEA, were both decreased. And again, so this time now carnosine was decreased and histidine increased, which is the reciprocal of the acute situation. Biotin was decreased. There's an increase in collagen turnover. There was a decrease in branched chain amino acids, plasmalogens. And I guess the main thing I want to bring out here is the decrease, again, continued decrease in purines, glycosphingolipids, and sphingomyelins that are also things that we've seen in chronic fatigue syndrome. So to turn quickly to the exposomics, so this is reporting on 207 of the 1,232 chemicals. And what we can say is that 80 to 100%, and in fact, this number is now actually 100% of both males and females were positive for at least one phthalate, at least one Teflon, and at least one pyrethrin. So these are universally found in the exposome in this study. And in addition, there are 47 other environmental chemicals that were seen in at least one of the samples. The others were negative, but a key question that we wanted to answer is the exposome of Lyme disease different in different regions of the country? And so far, there were no differences. So to summarize our results, again, we focused on the metabolomic analysis in females. And so, but only after first showing that males and females had a different but non-identical, so overlapping but non-identical metabolic response to Borrelia. So an acute Lyme showed a metabolic signature of the cell danger response, but plus some Lyme-associated things that appeared to relate to a portal of entry through the skin as a novel, it's a different way of introducing pathogens compared to through inhalation. So we had specific decreases in the 7-dehydrocholesterol, the immediate precursor of vitamin D, as well as the activated dihydroxy form of vitamin D3, a decrease in retinol, pyrophosphate, creatine, cysteamine, and histidine, and an increase in aldosterone, thyroxine, carnosine, and methylcysteine. Now, the convalescent Lyme was not the same as fully recovered and back to baseline. So multiple similarities to ME/CFS were found, including decreased purines, glycosphingolipids, ceramides, and cholesterol metabolism. And the exposome basically showed, with only a sixth of the chemicals so far completed the analysis, that Teflons, phthalates, and pyrethrins were present in 100% of the samples. And the exposome of cases and controls was the same so far in this pilot study. So this is a slide to thank our financial supporters, include the UCSD Christini Fund, the Stephen and Alexandra Cohen Foundation, the Coastal Foundation. And these particular samples that I'm talking about came from the Bay Area Lyme Foundation Biobank, but we have a larger study of over 400 samples that come from a collaboration with Johns Hopkins Lyme Biobank and Columbia Biobank as well. So I thank you and hope you have a great meeting.